Currently, we are in the unit 2. So we are discussing about.
Sorry, it is very
this.
fast, sir.
Huh?
Sorry, it is very fast, sir.
It is very first. I know. I'm going. So I will go directly to the point. So in the second unit, we have fungal diseases. So we already finished the superficial mycosis. Then we came to the second fungal disease, which is subcutaneous mycosis. So I will directly go to the subcutaneous mycosis. So I'm not going to discuss all these basics. We already know all of these things, right? We discussed everything. Let me move forward. We discussed a lot since week. Okay. It's a long story. Okay. So then we came to the unit 2. In the unit 2, we have this subcutaneous mycosis. So subcutaneous mycosis, that means these are the fungal infections that will cause infection to the subcutaneous layer of the skin. That is why they are called as subcutaneous fungal infections. Fungal infections, we are called them as a mycosis. Mycosis means fungal infections. So in the subcutaneous mycosis, we have 4 diseases. One is mycetoma. Second one is chromomycosis. Third one is sporotrichosis. And fourth one is rhinosporodiosis. So these are the 4 subcutaneous mycosis we have. So what are those? Mycetoma, chromomycosis, sporotrichosis, and rhinosporodiosis. So in the last lecture, we discussed in detail about mycetoma. Anyway, I will do a quick revision on mycetoma, then we can move forward. So mycetoma means this is typically a chronic granulomatous infection to the skin, which was very common in the South India, especially in the Madurai of Tamil Nadu. So this was first described by Gill, whereas Carter in 1860 established the fungal origin of the disease. Classically, this is called as Madura foot. This particular infection is called Madura foot, and it is very common in the tropical regions of the planet Earth. Okay, so it is it is a common tropical fungal infection, which is a chronic granulomatous infection to the subcutaneous tissue. Usually, this will confine to the foot. So usually, we will see this infection on the foot, but sometimes this infection can also spread into the other parts of the body other than the foot. Such infection is called eumycetoma. Eumycetoma will cause infection in any parts of the body, whereas actinomycetoma will only specifically target the foot of the person. Okay. So this is how a mycetoma infection looks like. So if you see the pathogenesis of mycetoma, the pathogenesis starts with a minor cut in the skin. Through this minor cut, the fungal spores will land into the minor cut. Then it will cause a localized swelling. Upon localized swelling, it will enlarge. It will make a tumor. The enlarged area, from this enlarged area, the fungus will start making. It will start digging deep into the tissue. It will start burrowing deep into the tissue, resulting in formation of abscess. So there will be multiple abscess on the surface of skin with multiple sinuses. Sinuses means these are like empty air spaces or holes within the tissue. So from these sinuses, it will start discharging viscid purulent pus. Okay. In this pus, we will see different types of granules. Upon closely examining the color of these granules, we can able to identify what type of mycetoma infection it is. So this is the typical pathogenesis of the mycetoma. Okay, it started with a abscess or a cut on or a small wound on the foot. From this small wound, the spores entered, it caused swelling, it caused tumor formation, or it enlarged, then it burrowed deep into the tissue, it prepared abscess, it prepared sinuses, and it started discharging mucopurulent viscid pus from the abscess. Okay, so this is the pathogenesis of the mycetoma. Then there are 2 types of mycetoma. As I already discussed, we have eumycetoma and actinomycetoma. So the species of eumycetoma is Madurella mycetomatis, which sheds black color spores from the pus or from the wound, whereas actinomycetoma, which is a classical mycetoma infection where we will see the wound on a foot. So this was occurred by 4 species. Those species include Actinomadura maduri, A. pelletieri, Nocardia brasiliensis, and Streptomyces somaliensis are the species that will cause actinomycetoma. Okay. So actinomadurai will discharge whitish yellow granules, whereas A. pilleti will discharge red granules. Nocardia brasiliensis will discharge white granules, whereas Streptococcus homilensis will discharge yellow color granules from the site of infection. So the laboratory diagnosis of this mycetoma typically involves observation of the granules present in this pus. So by observing these granules, we can able to say the type of fungal infection. So in the KOH preparation, we can able to identify chlamydia spores. So septate hyphae and chlamydia spores will present in the infection. Okay, in the KOH, we can able to see chlamydia spores. Okay, so these are few diagrams of the various species of the mycetoma. So this is how a acrimonium falciform looks like. This is how a microtrichophyton. Okay, this is how Streptomyces homilensis looks like, and this is how Actinomadura pelletieri looks like. Okay, so these are the few pictures and the fungus that causes mycetoma infection. Okay. Then coming to the treatment of mycetoma, this mycetoma, usually there is no treatment for mycetoma. If the infection is so severe, then we should proceed with amputation. Amputation means we need to cut the hand or infected area, the foot. Okay. Sometimes this mycetoma will respond to sulfonamides antibiotics. Okay. Few mycetoma species will respond to sulfonamides and antibiotics, but most of the time mycetoma requires amputation. That means we need to cut the area of infection. Okay, now that is all about this. So the examples of some antifungal agents include cotrimazole, streptomycin, or dapsone can be used for as an antifungal agent. Okay. Other than this, you can also use sulfonamides and antibiotics for treating this particular mycetoma infection. So this is all about the mycetoma. So after mycetoma, the second subcutaneous mycosis is chromomycosis. So let us see this chromomycosis in detail in today's lecture. So today I will try to finish chromomycosis and sporotrichosis. Okay. So let us move forward to the second subcutaneous mycosis, which is chromomycosis. Are you following me, students? Is there any doubts? No, right? No. Fine. So moving forward with chromomycosis. So what is this chromomycosis? Okay. So before this is how the chromomycosis infection looks like, students. So this is a typical chromomycotic infection. So in the chromomycosis, you will see cauliflower-like florets of tumors on the surface of skin. You will see like a cauliflower-like blobs on the surface of skin. Okay, this is a characteristic feature to identify the chromomycotic infection. Okay, so it is also a chronic localized infection of skin of the subcutaneous tissue. The fungus enters through a wound. So most of the subcutaneous fungus enters through wounds, it seems. So this fungus also enters into the body through wound. Etiological agent. Etiological means, keep in mind, student, this is a new word for you. Etiological means the meaning of etiology means origin or the cause. The causative agent is called etiology. Okay. So etiological agent. The disease causing agent is Fonsesia compactum, Fonsesia pedrosi, and two other species. At least try to remember these two fungus. So chromomycosis infection occurs due to Fonsesia pedrosi and Fonsesia compactum. So these two fungi are responsible for chromomycotic infection. Okay, so this name is Fonsesia pedrosi and other one is Fonsesia compactum. So both are darkly pigmented fungi that belongs to the Demataceae. So this taxonomy you need not to worry. You just remember this chromomycosis is occurred by a fungi called Fonsesia pedrosi and Fonsesia compactum. Fonsesia pedrosa and Fonsesia compacta are responsible for chromomycotic infection, which is a chronic granulomatous localized skin infection. So what type of skin infection? Subcutaneous tissue. Okay, subcutaneous skin infection. So the lesions caused by this chromomycosis looks like a warty cutaneous nodules. So they will look like a warty cutaneous nodules. Okay, resembling like a cauliflower. The nodules that are formed by this chromomycotic infection looks like a cauliflower-like nodules on the surface of skin. Okay, so The lab diagnosis of chromomycosis include, you can go with KOH, H&E staining, and in the KOH you will see various types of morphology that I will show. Yeah, in the KOH preparation, you can able to identify sclerotic bodies. So presence of sclerotic bodies is a characteristic feature to identify this particular chromomycotic infection. Okay, this sclerotic bodies can be seen in the biopsy specimens of the patient tissue. So you will take a tumor, a small piece of the patient tumor, then you will observe under microscope with H&E staining. Whenever you are taking biopsy, we will go with H&E staining, right? So upon H&E staining, we can able to see this sclerotic bodies. So presence of sclerotic bodies is a characteristic feature to identify this chromomycotic infection. Okay, so these sclerotic bodies can be either demonstrated in the KOH, or they can be demonstrated in the H&E staining, or they can also be seen in the cultures of STA. So in 3 of the cases, we will find this sclerotic bodies. So presence of sclerotic bodies is an identifying feature uh, uh, for finalizing that the patient has chromomycotic infection. Okay, then treatment for chromomycosis is amphotericin B and 5-fluorocysteine can be used as a they are the drugs of choice for treating this chromomycosis. So the treatment include amphotericin B and 5-fluorocysteine. Okay, amphotericin B and 5-fluorocysteine can be used as a drugs for treating this particular chromomycotic infection. So this is all about chromomycosis. So very simple, only 4 points. First one is chromomycosis is a chronic granulomatous subcutaneous tissue infection. Then second one, it is caused by Fonseca pedrosi, Fonseca compactum. Third one, it will cause cauliflower-like nodules on the surface of skin. Fourth one, presence of sclerotic bodies is a characteristic feature of chromomycosis. These sclerotic bodies can be identified by a KOH, SDA, or by H&E staining. Then treatment of chromomycosis includes amphotericin B and 5-fluorocysteine can be used as a drugs of choice to treat this particular chromomycotic infection. So this is all about chromomycosis. A quick revision: it is caused. It is a chronic infection, chronic skin infection. It is caused by Fonseca pedrosa and Fonseca compactum. They will cause cauliflower-like nodules on the surface of skin. Sclerotic bodies will be present. These sclerotic bodies can be identified by KOH, SD on the SD agar or in the by H&E staining. Then the treatment includes amphotericin B and 5-fluorocysteine. So these are the main points that you need to remember. And this is the diagram of the sclerotic bodies is very important. So try to practice this diagram. Okay, the diagram of this sclerotic bodies. So this is all about chromomycosis. Okay. So any confusions in chromomycosis? Very simple fungi actually. Right? It's very simple. So you can go through this material later. So now I'm moving forward to the third subcutaneous infection, that is sporotrichosis. So the third subcutaneous infection is sporotrichosis. If you remember my starting introductory class on subcutaneous mycosis, I have already discussed. This sporotrichosis is also called as gardener's disease because this disease is very common among the gardeners who will pluck the flowers from the plants. This particular infection we will get through thorn prick injury. That means through thorn pricks injury we will get this infection. And who will do this who will get who are mostly prone to this infection means gardeners, because they are the one who is always working with plants. They are the one who will always pluck flowers. So there is a high chance of getting injured by this thorns of the plants, and they will get this particular disease, fungal disease called sporotrichosis. That is why this sporotrichosis is also called as gardener's disease, because it is very common in gardeners and farmers. Okay, then this fungi will spread in the body through lymphatic channels. So through lymphatic vessels, this fungi will start spreading in the body. Okay, that is why you will see this characteristic band, a line of sporulations on the surface of skin. You can see sporulation like nodules, sporulated nodules on the surface of skin on a line. You know, what is this line? This is actually our lymphatic vessel. This fungus is spreading through the lymphatic vessel. Okay, so this fungus will spread through the lymphatic vessels of the body. So let us see some characteristic features of the sporotrichosis. So sporotrichosis is a nodular ulcerating disease of skin and subcutaneous tissue. So it is a nodular ulcerating disease of skin and subcutaneous tissue. It usually affects hand and forearm. So it will usually affects hand and forearm. Why? Because most of the thorn prick injuries happens to hands. So there is a high chance of getting uh, limb infection. So particularly hands and forearm infection. Then the fungus gain access through thorn pricks or some injuries. So usually through needle pricks. Okay, thorn means that plant needles. So through that needle injury, we will get this infection or this fungus will enter into our body. And it is very common in gardeners and farmers. So this particular infection is very common in gardeners and farmers. So the fungus spreads through lymphatic vessels. Okay, the fungus will spread through lymphatic vessels, and it will stay in the regional lymph nodes. So first fungus will gain access into lymphatic vessel, and these are lymph nodes. So all lymph nodes was infected by sporotrichotic fungi. So that is why in all the lymph nodes you will see ulcers. Okay, and this disease is everywhere worldwide. It is present, and it is very common in USA particularly. Okay, so the causative agent of sporotrichosis, the fungus that is responsible for this sporotrichosis is Sporothrix kenki. Sporothrix kenki is a fungi that is responsible for this sporotrichotic infection. Sporothrix kenki is a fungi responsible for sporotrichosis infection. Now, this is first time we are seeing a dimorphic fungi in subcutaneous infections. Keep in mind, this is the first time we are encountering a dimorphic fungi in subcutaneous infection. Okay, so Sporothrix kenki is a dimorphic fungi. Sporothrix kenki is a dimorphic fungi. When we say dimorphic fungi, that means it has two morphologies. Di means two, morphic means morphology. That means it has two morphologies. How it will have two morphologies? Those two morphologies are dependent on the temperature. One will be around 25 degrees centigrade and other one will be at 37 degrees centigrade. So let us see what are the two morphologies of this fungi. So if you see the morphologies of this fungi, the mold form will have flower-like appearance. The mold form will have flower-like appearance. Okay, flower-like clusters of conidia present in the mold form. Whereas east face, you will see cigar-shaped cells. Cigar-shaped cells are present in the east face, whereas flower-like flower-like conidia present in the mold form. Okay, so as you already know, east face will be seen around 37 degrees centigrade, whereas mold form will be seen at around 25 degrees centigrade. Okay, so uh, by by visualizing these two forms, we can able to conclude that it is a sporotrichotic infection. Okay, because it is a dimorphic fungi. So diagnosis is made by culture, as a fungi may not be demonstrable in the pus or tissues. So obviously we need to do culturing of this fungus, and we should grow this SD agar media in two temperatures. We need to incubate at 37 degrees centigrade, and also we need to incubate at uh, 25 degrees centigrade. At 25 degrees centigrade, you will see mycelial or mold form, whereas in 37 degrees centigrade, you will see yeast form. So the yeast cells appear like a cigar-shaped cells. So yeast cells looks like a cigar-shaped cells, whereas mold cells, the mold form will looks like a flower-like clusters. You will see flower-like clusters with sterigmata, with a very delicate sterigmata. You will see flower-like structure with a very delicate sterigmata in the mycelial form. So this is the identifying features of Sporothrix kenki. Yeast cells are in cigar shape, whereas mycelial phase has flower-like appearance with delicate sterigmata. Okay, so this fungus produces progressive disease in rats and interperitoneal or intertesticular inoculation. Okay, this will also cause infection in the rats. Okay, this is all about sporotrichosis. So a quick review of sporotrichosis. Sporotrichosis is a nodular ulcerating disease of skin, usually subcutaneous tissue of the body. It only affects hands and forearms. It will be gain access into body through thorn prick injury, which is very common in farmers and gardeners. This fungus will spread through the lymphatic vessels, and this is present worldwide, and mostly this is common in USA. The costly agents of sporotrichosis is Sporotrix kenki, which is a rhinosporotic fungi. So in the stem of this fungi, the mycelial form will looks like a flower with flowers with a delicate stigmata, whereas yeast form will have cigar-shaped yeast cells. Okay. That's it. This is all about sporotrichosis. Any doubts, students? Are you listening me?
Yes,
Yes, sir.
Yeah.
Yes, sir.
See, actually in the textbook there is no these images also. So in the textbook it is very straightforward without images. There you will feel even more boring. So I was trying my level best to keep it more interactive with this subject. I know this is slightly boring, but this is how we can learn this. Okay. Everything is new words. That's it. All you need to do is you need to memorize these fungus names, Sporotrix kenki or something like this. Okay. These names we can't escape. As a medical student, you need to by heart this bacteria or these names, fungal names. Then last and final subcutaneous infection is Rhinosporodiosis. As I said, Rhinosporodiosis means it is an fungal infection to the nose. Rhino or rhino means nose. Okay, the nose in the Greek it is called rhino. So it is a nose spores forming nose fungal infection. That is the meaning of the name. It is a spores forming nose fungal infection. Rhinosporodiosis. Okay, so this rhinosporodiosis is occurred by a fungi called Rhinosporidium seberi. Rhinosporidium seberi is a causative agent of rhinosporodiosis. This fungi will cause rhinosporodiotic infection. Okay, so this is a chronic granulomatous disease characterized by formation of febrile polyps. This particular word is very important. This rhinosporodiosis will cause this febrile polyps. These febrile polyps can be present in the nose or in the eyes. Okay, these polyps are called febrile polyps. These polyps can be present in the nose, mouth, eyes, and mucous membranes of the body. Okay, so it will cause febrile polyps in nose, mouth, and eyes and mucous membranes of the body. So 80% of rhinosporodiotic cases has been seen in India and Sri Lanka. That means it is very common in India and Sri Lanka. So 80% of cases are registered from India and Sri Lanka. So the problem with rhinosporodiosis is we cannot cultivate it. We cannot culture this particular fungi. That makes it very difficult to identify this particular infection since it is uncultivatable. Okay, so we cannot cultivate this fungi. Then how we can demonstrate this fungal disease? This fungal disease can be demonstrated by taking a biopsy of this febrile polyp. What's the proofs for this one, sir? Workshops, no, appraisal form. I think I'm not the one, sir. Yeah. Oh, yes, sir. So 20 minutes, sir, 20 minutes. Fine, sir. Oh, sorry, students. Okay, so how to identify this rhinosporodiosis? So this rhinosporodiosis can be identified by doing a biopsy. So you will take a small piece of this febrile polyp and you will do a section. And in the section, you will find sporangiospores, sporangium with endospores in that tissue. So presence of this endospores and sporangium is a characteristic feature to identify rhinosporodic infection. Okay, I will show you that sporangium and endospores. So yeah, where is the picture? This one. So in the tissues, we will find a large sporangium. So this circle is a sporangium actually. In this sporangium, you can see numerous sporangiospores. These sporangiospores are called as endospores. And you can see arteries, veins, and some tissues and cells. So among the cells, you will see this yeast cell. In this yeast cell, you can able to see sporangium with endospores. So presence of this sporangium is only identifying feature to conclude it is a rhinosporodic infection. Okay, but by the way, these endospores will be in millions. So you will see thousands of endospores within this sporangium. So presence of this endospores is a identifying feature for rhinosporodiosis. Coming to its properties, it is sporangium is around 10 to 200 micrometers in diameter, whereas endospores, each endospore is around 6 to 7 micrometers in diameter. So this is all about rhinosporodiosis. So after this, we can move on to the systemic mycosis. So from next week onwards, we can move on to the systemic mycosis. So this is all about the subcutaneous mycosis. A quick revision. In today's lecture, we discussed about chromomycosis. Chromomycosis is a chronic localized skin infection to the subcutaneous tissue. It is caused by Fonsesia pedrosi and Fonsesia compactum. This will cause cauliflower-shaped um, uh, tumors on the surface of the skin. Upon KOH and HND staining, we will see septate hyphae. Presence of sclerotic bodies is an identifying feature for chromomycosis. The treatment includes amphotericin B and 5-fluorocysteine. So these are the major points in chromomycosis. Then second one is sporotrichosis. So whenever I'm saying sporotrichosis, immediately you should get this word called. It is a Gardner's disease. It is a farmer's disease. It will cause ulcers which will spread through the lymph nodes of the body. This is caused by a fungi called Sporothrix kunki, which is a dimorphic fungi. And also just remember, sporotrichosis is a farmer's disease, which is caused by thorn prick injury. Hence, the fungus also looks like a flower. Since it is dimorphic fungi, the mycelial form looks like a flower with delicate sterigmata. So this is how you can able to identify the sporotrichotic infection. Coming to the rhinosporodiosis, it will cause febrile polyps in the eyes, mouth, and mucous membranes of the body. This febrile polyps cannot, this fungi, the causative agent is Rhinosporidium seberi. Unfortunately, we cannot cultivate this particular fungi. Rather than we will go for biopsy studies. In the biopsy, we can able to identify sporangium with thousands of sporangiospores or endospores within the sporangium. Upon identifying this, we can able to conclude it is a rhinosporodiotic infection. 80% of cases has been reported in India and Sri Lanka. That's it. So this is all about the subcutaneous mycosis. Okay. So a quick revision of this week's lecture. So in this week, we discussed in detail about subcutaneous mycosis, not only subcutaneous mycosis. We also discussed about dermatophytes and we discussed about subcutaneous mycosis. So I will do a quick overview. So all the students, are you listening? Students, are you there?
Yes, sir. Yes, sir.
Okay.
Yes, sir.
A quick interactive session and I will end this session. Okay. Um, which fungus is called Madurai foot? Which infection is called Madurai foot infection? Among these four.
Mycetoma, sir.
Mycetoma. Very good.
Mycetoma.
Mm, then what is the difference between eumycetoma and actinomycetoma? Difference between eumycetoma and actinomycetoma. Foot infection is caused by what type of mycetoma? Is it mycetoma or actinomycetoma? Foot infection. Foot infection is caused by what type of mycetoma? Actino or U?
Sir, actino.
Actinomycetoma, very good. Actinomycetoma will cause foot infection, whereas mycetoma will cause body infection. Then, what fungal infection is called Gardner's disease? What fungal infection is called Gardner's disease? Tell me, students.
Sir, Sporotrichosis.
Sporotrichosis.
Very good. Sporotrichosis. What fungus will cause sporotrichosis? Fungus name?
Fungus name is sir.
Sir, big name, sir.
Simple name.
Sporotrix.
Skanky. Okay. Sporotrix skanky. Okay.
Sporotrix skanky.
Skanky. Okay. Sporotrix skanky. And among these 4 diseases, which one is dimorphic fungi? Among these 4, which fungus
Sir,
is a
garden,
dimorphic?
sir. Garden is, sir.
Yes, which
Yes,
is
sir.
sporotrichosis. Same. You haven't changed it. Okay, then what fungus infection we will see sclerotic bodies? In what fungal infection we will see sclerotic bodies?
Chromomycosis.
Chromomycosis, very good. What is the fungus name? Chromomycosis is caused by what fungi? What is the name of fungi? See, it will be good if you prepare a table. Okay, so prepare a table like this. So draw the 4 lines and write fungus names. Mycetoma is by 4 species. Then chromomycosis is caused by Fonseca pedrosa and Fonseca compactum. Sporotrichosis is caused by Sporotrix skenki, whereas Rhinosporodiosis is caused by Sebri. So you just make a table for 4 diseases in 1 page. Then you can able to easily memorize it. Okay. Among these 4 fungi, which will spread through lymphatic vessels? What fungus will spread through lymphatic vessels?
Sir, Sporotrichosis.
Sporotrichosis. Very good. Very good. Then what fungus we cannot cultivate? We are unable to cultivate in the lab. Which fungus is unable to cultivate in the lab?
Rhinosporodiosis. Sporodiosis.
Rhinosporodiosis. How you will identify rhinosporodiosis? By seeing
Through biopsy.
sporangium with endospore. Very good. By biopsy. Very good. Then, what fungus is most prevalent in Tamil Nadu? In Madurai?
Sir, mycetoma, sir.
Mycetoma. And then Sri Lanka and India? What fungus is more common in Sri Lanka and India?
Rhinosporodiosis.
Are you sure?
Yes, sir.
Very good. Then what fungus infection is very common in USA? I discussed. This particular fungi is common in USA. Sporotrichosis. Very good. Then what are sclerotic bodies? What are sclerotic bodies?
Chromomycosis.
Okay, sclerotic bodies are specialized cells that can be observed in the chromomycotic infection. Tell me the fungus name of chromomycosis. There are 2 fungi. Fonseca pedrosa and Fonseca compactum. I remember the name, students. It seems like I'm learning again. Okay, so just practice this, then you will get it. Okay, kindly start preparing notes and go through the notes once. See, one important thing in mycology is you need repeated revisions. At least once a week you need to do revision. If not, you will confuse. You will write one example for another infection. You will write Francisca pedrosi for sporotrichosis. You will write dermatophytal examples in systemic infections. So in order to avoid that confusion, you need to do thorough revision, at least 10 minutes a day. Just 10 minutes a day is sufficient for revision. Just go through the notes once. Prepare the notes and go through the examples and names. That's it. Without revisions, your brain will become fry. You will become confused. Okay. So this is what I can say. So thank you very much, students. You can leave. So from tomorrow onwards, from next week onwards, we can start systemic mycosis. Then we can say goodbye to mycology. Then we can start our actual classes on virology. Okay. So thank you very much, students. I'm ending this session.
Thank you, sir.
Yeah.